welcome to the deep dive. Today, we're really aiming for, let's say, mastery-level understanding of epithelial tissue.
Yes, it's fundamental stuff. Epithelium, it covers things, it lines things. Seems simple, right? But getting that true mastery means digging into the molecular details, the architecture, how these cells form this incredibly selective barrier that controls basically everything moving in and out of body compartments.
So we're going way past just definitions today.
Absolutely. We need to see how they maintain that control.
Okay, context first. The body seems infinitely complex, but it's built from just 4 basic tissue types.
It's kind of amazing, isn't it? You've got connective tissue, muscle tissue, nervous tissue, and then our focus today, epithelial tissue.
So connective tissue, that's mostly about support and lots of extracellular matrix, the stuff between cells.
Exactly. ECM is abundant there. Then muscle tissue, it's all about those long contractile cells for movement.
And nervous tissue, long processes again, but for sending signals.
Right, transmitting those electrochemical impulses.
Which makes epithelia the odd one out, in a way.
It really is. The defining feature is how tightly packed the cells are. Polyhedral shapes, really close together.
Leaving almost no room for that extracellular matrix.
Precisely. They form these continuous sheets, covering surfaces, lining cavities. It's a different strategy entirely.
And that structure dictates their main jobs.
Absolutely. Their mission, if you like, covering and protecting surfaces, think skin.
Okay.
Absorption, the lining of your gut, is a prime example.
Right.
And secretion, that's what glands are made of.
But it gets even more specialized, doesn't it?
Okay.
You mentioned some niche roles.
Oh, definitely. Some epithelial cells become sensory receptors, like in taste buds or your olfactory system, detecting smell.
Sensory, okay.
And then you have these fascinating cells called myoepithelial cells. They're epithelial, but they can contract, like muscle. They help squeeze secretions out of glands.
A contractile barrier cell. That's interesting.
It is. And this idea of function leads us to a really important concept in histology, parenchyma versus stroma.
Ah, yes. How organs are organized.
Right. The parenchyma is the collection of cells that performs the main function of the organ, the specialized work.
And in most organs, that's the epithelial cells.
Overwhelmingly, yes. The epithelial cells are the parenchyma.
So what's the stroma then?
The stroma is the support crew. It's a connective tissue framework that holds everything together, provides the blood supply, the nerves.
Got it. Always connective tissue.
Almost always. The big exception is the brain and spinal cord, where glial cells form the support structure. But elsewhere, stroma means connective tissue support for the epithelial parenchyma. It's a key division of labor.
Okay, let's dive into the cells themselves. Their shape, you said it's dictated by function. Squamous, cuboidal, columnar?
Exactly. Squamous are flat, like tiles. Cuboidal are cube-shaped, roughly equal height and width. Columnar are tall, like columns.
And the nucleus shape usually matches.
Generally, yes. Flat nucleus in squamous, round in cuboidal, elongated in columnar.
And since the cell membranes are hard to see with a basic light microscope,
The nucleus becomes our main guide. Its shape, its position, how many layers of nuclei you see that tells you about the cell shape and the tissue structure.
now the concept that always comes up is polarity. You mentioned it briefly. Why is this uneven distribution inside the cell so critical?
It's everything, really. Polarity means the cell has a top and a bottom, or maybe a front and back, that are structurally and functionally different. It has to be this way to do its job.
Like absorbing on one side and secreting on the other.
Precisely. Or having different channels and pumps on different surfaces. We talk about 3 main surfaces or poles.
Okay, what are they?
There's the basal pole, that's the bottom surface, the one sitting on the underlying connective tissue and its extracellular matrix.
Right, the base.
Then there's the apical pole. This usually faces outwards into a lumen or a free space. This is often where you find specialized structures like microvilli or cilia.
The working end, maybe?
You could think of it that way, yeah. And finally, you have the lateral surfaces. These are the sides where the cell connects to its neighbors.
And these aren't just flat walls.
Often not. They can have lots of folds and interdigitations to increase the surface area for cell-to-cell connections and communication. So the cell is compartmentalized, functionally speaking.
Okay, so you have this organized cell sitting on connective tissue. But you mentioned something crucial. Epithelia are normally avascular, no blood vessels.
Correct. A huge point.
That sounds like a major problem. How do they get nutrients? How do they get oxygen? Especially if they're doing energy-intensive work like absorption or secretion.
It all comes down to diffusion. Everything they need, oxygen, nutrients, has to diffuse across from the capillaries located in that underlying connective tissue.
So that interface between the epithelium and the connective tissue is absolutely critical.
And life or death for the epithelial cells, which is why that supporting connective tissue often has a specific name. In places like the gut, respiratory tract, urinary system, we call it the lamina propria.
The lamina propria, that's the supply line.
Exactly. And to make that supply line even more efficient, especially in areas subject to friction, like your skin or tongue,
the connective tissue often pushes up these little finger-like projections into the epithelium. They're called papillae.
like bumps?
Kind of, yeah.
Yeah.
They dramatically increase the surface area of contact between the vascular connective tissue and the avascular epithelium, ensures better nutrient delivery.
Makes sense. This close relationship naturally brings us to the basement membrane, the BM.
Yes, the foundation itself. It's this thin, sort of felt-like sheet of extracellular material that the basal surface of every epithelium rests upon.
It's the anchor point, but you also said it's a filter.
A critical filter, yes.
Yeah.
Semi-permeable. It sits between the connective tissue capillaries and the epithelial cells, regulating what gets through.
Now, I know the terminology here can be tricky. Basement membrane, basal lamina.
It can be confusing.
Huh?
Under a regular light microscope, you just see one layer, the basement membrane, often stained with PA's technique.
Okay.
But zoom in with an electron microscope, and you see it's actually two distinct layers.
2 layers, what are they? First, right up against the epithelial cells is the basal lamina, or BL. It's thin, maybe 20 to 100 nanometers, and looks dense in electron micrographs.
And the key thing about the basal lamina...
It's secreted by the epithelial cells themselves. They build their own immediate foundation.
Okay, epithelial cells make the BL. What's the second layer?
Beneath the BL is the reticular lamina, or RL. This layer is usually thicker, more diffuse, more fibrous looking.
And who makes the RL?
That's made by the cells in the underlying connective tissue, mainly the fibroblasts, so the complete basement membrane is really a joint project.
A collaboration between epithelium and connective tissue. Let's get molecular with the basal lamina. What's it made of?
It's a beautiful piece of engineering. The core scaffold is type IV collagen. Unlike other collagens that form fibers, type IV forms a meshwork, like a window screen, a 2D lattice.
A mesh. Okay.
Yeah.
How does the cell attach to that mesh?
Through large glycoproteins called laminins. These are multi-adhesive proteins. One end attaches to transmembrane proteins in the epithelial cell membrane called integrins.
Integrins in the cell membrane.
And the other parts of the laminin molecule weave through and bind to that type of V collagen mesh, so laminin is the bridge.
Got it. Cell connects to integrin, integrin connects to laminin, laminin connects to the type 4 mesh. What holds the mesh together and gives it its 3D structure?
That's where other proteins come in, primarily nitogen and perlecan. Nitogen is sort of rod-like. Perlecan is a proteoglycan. They act as cross-linkers.
Cross-linkers.
Yeah, they link the laminins to the type 5e collagen network. They stabilize the whole structure. And crucially, their arrangement helps determine the porosity of the basal lamina.
Ah, so they control the filtering function.
Exactly. The size and charge selectivity of the filter depends on how these components are organized. Hugely important in the kidney glomerulus, for example.
Okay, that's the BL. What about the reticular lamina underneath? What's anchoring that?
The RL mainly contains type 3 collagen, which forms reticular fibers, and it's securely attached to the basal lamina above it by special anchoring fibrils made of type 7 collagen.
Type 7 anchoring fibrils. Okay. So the BM is much more than just a physical support.
Oh, much more. It provides structural support, yes. It filters, it helps maintain that crucial cell polarity we talked about.
How does it do that?
By binding specific receptors on the basal surface, it helps organize the internal cytoskeleton and keep things in their proper place. It also localizes signaling molecules.
Okay.
And very importantly, during tissue repair, the BM acts as a scaffold. It guides migrating epithelial cells so they regenerate in an organized way. It's essential for effective healing.
Right, so it's structure, filter, organizer, and repair scaffold. That's quite a list.
It's a fundamentally important structure.
Okay, we've anchored the base. Let's move up to the sides, the lateral surfaces. How do these cells stick together so tightly, especially under stress?
That's the job of the junctional complexes. These are specialized spots on the cell membrane that handle adhesion, sealing the gaps between cells, and even allowing communication.
3 main jobs: sealing, sticking, communicating. Let's start with the seal.
That would be the tight junctions. The proper name is zonulae occludens.
Zonula meaning belt.
Exactly. It forms a continuous band, like a belt. encircling the very top, the apical end of each cell. It looks like the membranes are literally fused together there.
And the key proteins involved?
Two main families, claudins and occludins. These transmembrane proteins stitch the adjacent cell membranes together.
Okay, claudins and occludins. What does this seal actually do? You said two key roles.
Right. First, it creates the paracellular barrier. It blocks stuff from just flowing freely between the cells.
So it forces molecules to go through the cell instead, the transellular path.
Precisely. It forces controlled, often energy-dependent transport through the cell cytoplasm. And how tight the seal is varies.
How so?
It depends on the number of sealing strands formed by the claudins and occludins. Your urinary bladder lining needs to be super impermeable, so it has many strands. A kidney tubule, which does allow some controlled paracellular movement, has fewer.
Okay, that's the barrier function. What's the second role? The fence.
The fence function is about maintaining polarity. This junction literally prevents membrane proteins and lipids that belong on the apical surface from drifting down into the basolateral domain and vice versa.
It keeps the top proteins at the top and the bottom proteins at the bottom.
Exactly. Without that fence, the specialized functions of the apical and basolateral membranes would just blur together, and the cell couldn't do its job. Polarity would be lost.
And these junctions are vulnerable. You mentioned pathology.
Very much so.
Yeah.
Certain bacterial toxins specifically target these proteins, like the enterotoxin from Clostridium perfringens, the food poisoning bug.
What does it do?
It messes with claudin assembly. It prevents the seal from forming properly. And the result, massive fluid loss between the cells into the gut. Diarrhea.
Wow.
Similarly, Helicobacter pylori, the ulcer bug, disrupts signaling related to these junction proteins. It shows how critical this molecular seal is for our health. Defects in occludins can even affect the blood-brain barrier and development.
Okay, tight junctions seal the top. What's immediately below them?
Typically, you find the first major anchoring junction, the adherent junction, or zonula adherens, another belt-like structure.
Zonula again, so it goes all the way around.
Often, yes. It sits just below the tight junction and helps stabilize it, providing mechanical strength.
What proteins are doing the sticking here?
These rely on cadherens, a big family of transmembrane proteins that need calcium ions to function. That's where the cad in cadherin comes from. They bind to identical cadherens on the neighboring cell.
Adherens. Calcium dependent. And inside the cell, what do they connect to?
Inside, the cadherens link up with proteins called catenins.
Catenins.
And the catenins then connect the whole junctional complex to the cell's internal skeleton, specifically to the network of actin filaments.
Actin. Okay, so tight junctions seal, adherens junctions anchor to actin.
Right, and that actin network they connect to is part of what's called the terminal web, that reinforcement layer just under the apical membrane.
Is there a clinical link here too?
A huge one in cancer biology. Many aggressive epithelial cancers, called carcinomas, show a loss or downregulation of E-cadherin.
E-cadherin, a specific type.
Yes. And when cells lose that E-cadherin-mediated adhesion, they become less stuck together. It makes it easier for them to detach from the primary tumor, invade surrounding tissues, and metastasize. It's a key step in cancer progression.
Loss of adhesion enables spread.
Okay, next anchoring junction.
The powerhouse of adhesion. The desmosome or macula adherens.
Macula means spot, so this isn't a belt.
Correct. Desmosomes are like spot welds, disc-shaped, incredibly strong points of attachment scattered across the lateral surfaces, providing strength against shearing forces.
Stronger than adherens junctions. What makes them so tough?
They use different cadherin family members, desmoglanes and desmocolins. These link internally to a very dense plaque of proteins, including plicoglobins and desmoplackins.
Okay, different cadherins, dense plaque. But what's the real key to their strength? It's what they connect to inside the cell. The desmosomal plaque via desmoplackins links not to actin, but to the much thicker, rope-like intermediate filaments.
Intermediate filaments,
Well,
like keratin in epithelial cells.
Exactly. Cytokeratin filaments, sometimes called tonofilaments. These filaments are incredibly strong and distribute mechanical stress throughout the cytoplasm and across the entire tissue sheet. It makes the epithelium highly resistant to physical tearing.
And if these fail?
You get devastating blistering diseases. In conditions like pemphigus vulgaris, the patient makes autoantibodies against their own desmoglanes.
Antibodies attacking the desmosome protein.
Yes. The cell-to-cell adhesion weakens dramatically, the epithelial layers separate, and fluid accumulates, causing severe blisters on the skin and mucous membranes. It highlights just how vital that desmosomal connection is.
Wow. Okay, so we have seals, tight junctions, and strong anchors, adherens junctions and desmosomes. What about communication?
That's the role of the gap junction, or nexus. These are purely about letting adjacent cells talk to each other directly. They don't provide significant adhesion or sealing.
How do they work?
They're formed by clusters of transmembrane protein complexes called connexins. Each connexin is like a little barrel made of 6 individual connexin proteins.
6 connexins make one connexin.
Right, and when a connexin in one cell membrane lines up perfectly with a connexin in the adjacent cell membrane, they dock together.
Creating your channel.
Exactly. They form a continuous, regulated hydrophilic pore, about 1.5 nanometers wide, that directly connects the cytoplasm of the 2 cells.
What can pass through that pore?
Small molecules and ions. Things like cyclic AMP, calcium ions, metabolites. It allows for very rapid electrical and metabolic coupling between cells.
So they can coordinate their activity.
Absolutely. This is essential in tissues where cells need to act in unison, like the coordinated contractions of heart muscle or smooth muscle in the gut.
Any medical relevance?
Yes, mutations in connexin genes have been linked to certain forms of deafness because gap junctions are crucial for function in the inner ear and also some peripheral neuropathies.
Okay, so that covers the lateral junctions. Before we leave the base completely, what about anchoring the cell itself to the basement membrane? We mentioned the BM earlier.
Right, we need to connect the cell's internal skeleton to that external foundation. There are 2 main structures for this. First, hemidesmosomes.
Hemi meaning half, like half a desmosome.
They look a bit like half a desmosome ultrastructurally, yes, but molecularly they're completely different. They do not use cadherens.
No cadherens, what do they use?
They use integrins, those transmembrane receptors we mentioned earlier that bind to the basal lamina, specifically to laminin.
Okay, integrins bind to laminin in the BL and inside the cell.
Inside, the integrins in a hemidesmosome connect indirectly to the intermediate filaments, the cytokeratin network. So they provide a very strong, stable anchor linking the cytoskeleton to the BM.
Strong anchor to intermediate filaments. What's the second structure?
The second type are focal adhesions, or focal contacts. These are generally smaller, more numerous, and more dynamic than hemidesmosomes.
Dynamic, meaning they can change.
Yes, they're particularly important during cell movement, like when epithelial cells need to migrate during wound healing or tissue remodeling.
How are they different molecularly?
They also use integrins to connect to the basal lamina. But inside the cell, their integrins link primarily to bundles of actin filaments, not intermediate filaments.
Actin again, like adherens junctions.
Right. And importantly, focal adhesions are also major signaling hubs. The integrins connect to enzymes like focal adhesion kinase, or FAK, which triggers intracellular signaling pathways that regulate cell adhesion, migration, survival, and proliferation. They're anchors and sensors.
Okay, that covers the foundation and the sides. Let's move to the top edge, the apical modifications. These are the really specialized structures, right?
Absolutely. These are all about interacting with the outside world or the lumen, usually for absorption or movement.
Let's start with the most common one for absorption, microvilli.
Right. Microvilli are these short, finger-like projections of the cell membrane. Very uniform, typically about 1 micrometer long.
And when they're densely packed, like in the intestine.
They form what we see under the light microscope as a brush border or striated border. It looks like a fuzzy edge on the cells.
And their function is just surface area.
Primarily, yes. They can increase the absorptive surface area by an incredible amount, maybe 20 or 30 times. But there's more to it. The membrane covering the microvilli has a thick carbohydrate coat called the glycocalyx, and embedded within this glycocalyx and the membrane itself are critical digestive enzymes.
So they help with the final breakdown of food before absorption.
Exactly. They complete the digestive process right at the absorptive surface.
What's inside a microvillus? What holds it up?
The core is made of bundles of actin filaments. These are anchored to the very tip and sides of the microvillus membrane by various actin-binding proteins.
Actin again. And where do these actin bundles connect at the base?
They insert into that terminal web the network of actin and other cytoskeletal filaments just beneath the apical cell membrane. It provides support for the whole structure.
Are they scatic?
Not entirely. There's evidence for some myosin-based movement, which might help stir the local environment or maintain optimal spacing for absorption, that they're dynamic to some extent.
And if you lose them, we mentioned celiac disease.
Yes, a classic example. The autoimmune reaction triggered by gluten leads to inflammation and destruction of the intestinal villi and their microvilli brush border.
Resulting in?
Severe malabsorption, because you lose that massive surface area and the enzymes associated with it. It directly links the integrity of this tiny structure to the body's ability to get nutrients.
Okay. Next, apical structure. Stereocilia. Confusing name.
Very confusing name. They sound like cilia, but they're absolutely not related to cilia in structure or function. They're essentially very, very long microvilli.
Long microvilli, but still with an actin core.
Yes. Still have an actin filament core, still connected to the terminal web. They can be much longer than typical microvilli and sometimes branched. They're much less common.
Where do you find them? What do they do?
Two main places. In parts of the male reproductive tract, like the epididymis, they seem to function mainly in absorption, increasing surface area.
Okay, absorption. And the S-space.
In the inner ear. Here, they act as the primary sensory structures, the mechanoreceptors that detect sound vibrations and head movements. Bending of the stereocilia triggers the nerve impulse.
So, same structure, but radically different function depending on location. Fascinating.
Right.
Now for the real cilia.
Right. Cilia are fundamentally different. They're built for motility. They're longer than microvilli, maybe 5 to 10 micrometers, and significantly wider.
And the key difference is inside. No actin.
Correct. The core of a motohelium is made of microtubules, arranged in a very specific, highly conserved pattern.
Wow, the famous 9+2 structure.
Exactly. The axoneme is the name for this internal microtubule skeleton. It consists of 9 pairs, or doublets, of microtubules arranged in a circle around 2 single central microtubules. 9 peripheral doublets, 2 central singlets, 9+2.
And this structure is essential for movement.
Absolutely. How does it move? It involves motor proteins called dynein arms. These are attached to 1 microtubule in each outer doublet.
Dynein, like little motors.
Precisely. They use the energy from ATP hydrolysis to walk along the adjacent microtubule doublet.
Walk along it.
Yes, but because all the doublets are anchored at the base, they can't just slide past each other freely. This restricted sliding causes the whole axoneme to bend.
Ah, so the sliding causes bending.
Right. And the coordinated sequential bending of cilia creates a whip-like beating pattern that propels fluid or mucus-containing trapped particles across the surface of the epithelium. Think of the respiratory tract clearing debris.
That requires a lot of energy, a lot of ATP for all those dynein motors.
It's a highly active process. And where do these cilia anchor into the cell?
Good question.
The axoneme is continuous with a structure in the apical cytoplasm called the basal body. Basal bodies have essentially the same structure as centrioles, 9 triplets of microtubules, with no central pair. They organize the axoneme assembly and anchor it to the cytoskeleton.
Okay. Now, I've also heard of primary cilia. Are they different?
Yes. The primary cilium is a bit of an outlier. It's usually solitary, 1 per cell shorter, and critically, it's non-motile.
Non-motile.
Why? It lacks the central pair of microtubules, so it has a 9+0 structure, and it doesn't have the dynein arms needed for movement.
So what does it do?
It acts as a cellular antenna. It's loaded with receptors and ion channels, sensing the cell's environment, detecting flow, chemical signals, light, odor. It plays crucial roles in signaling pathways during development and tissue maintenance, found on almost all cell types.
A sensory antenna. Okay, and what happens if the motile cilia don't work properly?
That leads to conditions like immotile cilia syndrome, also known as primary ciliary dyskinesia or Cardiganer syndrome. It's usually caused by genetic mutations affecting proteins needed for cilia structure or function, like dynein.
And the consequences?
They're systemic. In the respiratory tract, the lack of mucus clearance leads to chronic sinusitis, bronchitis, and recurrent infections.
Makes sense.
And because the sperm flagellum uses the same 9+2 axoneme structure for motility, men with this condition are usually infertile due to immotile sperm. It really drives home how structure dictates function.
Absolutely. Okay, let's shift from the single cell level to classifying the entire epithelial sheet. How do we categorize them?
It's based on 2 straightforward criteria you can usually see with a light microscope. First, the number of cell layers. Is it just 1 layer thick, simple, or are there 2 or more layers, stratified?
Simple or stratified?
Okay.
And the second criteria?
The shape of the cells in the outermost layer, the layer facing the free surface or lumen. Are they squamous, cuboidal, or columnar?
Got it. Number of layers and shape of the top layer. Let's do simple epithelia first, 1 layer thick.
Right. Simple means optimized for exchange, transport, secretion, or absorption where you don't want a thick barrier.
So simple squamous, Flat cells, one layer.
Exactly. Very thin. Perfect for lining surfaces where rapid diffusion or filtration occurs. We find it lining blood vessels and lymphatics where it's called endothelium.
Endothelium, okay.
And lining the major body cavities like the pleura, pericardium, barytonium, where it's called mesothelium. It also allows organs to slide past each other smoothly. And these cells can be quite active in transport via penocytosis and secretion.
Next, simple cuboidal. One layer of cube-shaped cells.
Right. Thicker than squamous, so more cytoplasm, more organelles, especially mitochondria. This supports more active processes like secretion and active transport. Good examples are the surface of the ovary, or the tubules in the kidney, or thyroid follicles.
And simple columnar. Tall cells, one layer.
Yes, often highly polarized. Found lining the stomach, the intestines, the gallbladder. Frequently have apical specialisations like microvilli for absorption or cilia for movement. Their functions are protection, lubrication, absorption, secretion, real workhorse cells.
Okay, that's the simple types. Now, for stratified multiple layers, what's the main job here?
Protection. Durability. Stratified epithelia are found in areas subject to mechanical stress or abrasion. And remember, we classify them based on the shape of the topmost layer of cells.
Right, the surface layer shape is key. So stratified squamous is the most common.
By far. And it comes in two main flavors. First, stratified squamous keratinized.
Keratinized, like skin.
Exactly. That's the epidermis. The basal cells are cuboidal and actively dividing. As cells get pushed upwards, they differentiate, flatten out, accumulate huge amounts of the tough protein keratin, and eventually lose their nucleus and organelles to become dead, protective scales called squames.
Providing a tough, waterproof barrier.
Essential for preventing water loss and protecting against abrasion and microbes on dry surfaces. The other flavor is stratified squamous, non-keratinized.
Non-keratinized, so no dead layer on top.
Correct. This lines moist internal surfaces that still need protection from abrasion, like the inside of your mouth, the esophagus, the vagina. The surface cells are still living, nucleated squamous cells. It's protective, but not waterproof like skin.
Okay. Are there other stratified types?
Yes, but they're much rarer. Stratified cuboidal is usually just 2 or maybe 3 layers of cuboidal cells. You find it lining the larger ducts of sweat glands or salivary glands. Offers more robust lining than simple cuboidal.
And stratified columnar. Even rarer.
Very rare. Again, usually just 2 layers with columnar cells on the surface. Found in places like the conjunctiva lining your eyelids or the largest ducts of some glands. Primarily protective.
Now, there are a couple oddballs that don't fit neatly, right? Pseudostratified.
Ah, yes. Pseudostratified columnar epithelium. Pseudo meaning false. It looks stratified because the nuclei are all at different levels within the layer.
Looks stratified, but it isn't. How does that work?
The key is that all the cells, even the ones that look like they're at the bottom, actually rest on the basement membrane. However, not all of them reach the free surface, so it's technically a single layer, just a very crowded and messy-looking one.
Where do we find this?
The classic example is the lining of the upper respiratory tract, the trachea and bronchi. It's typically heavily ciliated and packed with mucus-secreting goblet cells. The respiratory epithelium.
A mucus escalator.
Okay.
And the other special type, transitional?
Transitional epithelium, also called urothelium, because it's exclusively found lining the urinary system, the renal pelvis, ureters, bladder, and parts of the urethra.
What's special about it?
It's built to stretch. When the bladder is empty, the epithelium looks like it has maybe 5 or 6 layers, and the surface cells are large, dome-shaped, sometimes even binucleated. We call these umbrella cells.
Umbrella cells.
Yes, they cover several underlying cells, but when the bladder fills with urine and stretches,
It flattens out.
Exactly. The epithelium thins down to maybe just 2 or 3 layers, and the umbrella cells flatten out considerably. This allows the organ to distend significantly without tearing the lining. Plus, those umbrella cells have a unique, specialized membrane that's impermeable to urine, protecting the underlying tissues.
Very specialized for its location. Now, this ability of epithelia to change brings up metaplasia. What is that?
Metaplasia is a reversible change, where one mature epithelial type is replaced by another mature epithelial type, usually in response to chronic irritation or stress.
Reversible, so it can change back.
Potentially, yes, if the stimulus is removed. A classic example occurs in the airways of heavy smokers.
What happens there?
The normal ciliated pseudostratified columnar epithelium, which is good at clearing mucus, gets replaced by stratified squamous epithelium.
Why would it do that?
Stratified squamous is tougher, more resistant to the physical and chemical irritation of smoke. It's a protective adaptation.
But there's a downside.
Big downside. You lose the cilia and the goblet cells, so you lose the mucus clearance mechanism, leading to increased risk of infection and potentially paving the way for cancerous changes down the line. It's adaptation, but at a functional cost. Chronic vitamin A deficiency can also cause metaplasia in some tissues.
Okay, let's switch gears to secretion. Glands are essentially epithelial tissues specialized for making and releasing stuff, right?
Exactly. Epithelial cells that are highly specialized for synthesis, storage, and release of macromolecules, proteins, lipids, carbs, or combinations.
That's the simplest kind of gland.
The unicellular gland, just single, isolated secretory cells scattered within a non-secretory epithelial lining.
And the prime example is?
The goblet cell. You find them everywhere in the lining of the intestine and the respiratory tract.
What do they secrete?
Mucus. Specifically, they produce mucins, which are heavily glycosylated proteins. When released and hydrated, they form mucus, which lubricates and protects the surface.
And they look like goblets, hence the name.
Pretty much. The apical part of the cell gets stuffed full of pale-staining mucinogen granules, making it bulge out, while the nucleus gets pushed down to the narrow base. They stain brightly with PAS because of all the carbohydrates in the mucin.
Oh, okay, that's unicellular. How do multicellular glands form?
They develop during embryogenesis from a covering epithelium. The epithelial cells proliferate and grow downwards, invading the underlying connective tissue.
And what happens next determines the type of gland.
Correct. If the connection to the surface epithelium is maintained in the form of a duct, then it's an exocrine gland. The secretions travel through the duct to reach the surface, either external, like sweat glands, or internal, like pancreatic enzymes, into the intestine.
Exocrine means secretion via duct to a surface. What if the connection is lost?
If the connection disappears during development, the gland becomes ductless. This is an endocrine gland.
No duct. So how do the secretions get out?
They secrete their products, which we call hormones, directly into the surrounding interstitial fluid. From there, the hormones are picked up by nearby capillaries and enter the bloodstream to travel throughout the body and act on distant target cells.
Endocrine is ductless secretion into the blood. Okay, let's focus on exocrine glands. How are they classified structurally? Seems complex.
It can be, but it's logical. We look at two things: the structure of the duct and the shape of the secretory portion.
Okay, ducts first.
Ducts can be simple, meaning the duct is unbranched, or they can be compound, meaning the duct branches one or more times.
Simple or compound duct. Got it. Now the secretory part.
The secretory units can be tubular shaped like a tube, which can be straight, coiled, or branched. Think intestinal glands or sweat glands.
Tubular.
Or they can be acinar, also called alveolar, shaped like a rounded sac or flask, often seen in glands producing more viscous secretions, like sebaceous glands or parts of the salivary glands.
Acinar means sac-like. Any others?
You can also have tubuloacinar glands, which have a mix of both tubular and acinar secretory units emptying into the duct system. Large glands, like the pancreas or major salivary glands, are often compound tubuloacinar.
So you combine the duct type and secretory shape, like simple coiled tubular or compound acinar.
Exactly. It describes the gland's architecture. Now, besides structure, we also classify exocrine glands by how they release their secretions. The mechanism.
Right, you mentioned three mechanisms earlier. Merocrine.
Merocrine is the most common. The secretory product is synthesized, packaged into membrane-bound vesicles or granules, moved to the apical surface, and released by endocytosis. The cell membrane fuses with the vesicle membrane, releasing the contents without losing any cytoplasm.
So the cell stays intact, like the pancreas releasing enzymes.
Precisely. Most protein-secreting glands work this way.
Okay, second mechanism, holocrine.
Holocrine secretion is the most dramatic. The cell fills up with its secretory product, and then the entire cell disintegrates, ruptures, releasing the product along with all the cellular debris into the gland's lumen.
The whole cell dies and breaks apart. Wow, where does that happen?
The classic example is the sebaceous glands in your skin that produce oily sebum. The cells literally self-destruct to release their lipid-rich product.
Which is why blocked sebaceous glands and acne get inflamed, all that cellular debris.
Exactly. It provides food for bacteria and contributes to the inflammation.
Okay. And the third mechanism, apicrine, kind of in between.
Apicrine secretion involves the product accumulating at the very apical tip of the cell. Then, a portion of that apical cytoplasm, membrane and all, gets pinched off and released along with the product.
So the cell loses a bit of itself but doesn't die.
Right. It survives and reforms. This mechanism is traditionally associated with lipid secretion in mammary glands, though some debate its prevalence elsewhere. It's distinct from merocrine and holocrine.
Merocrine exocytosis, holocrine cell death, apicrine pinching off.
Got it. Now, within the common merocrine glands, are there different types of secretory cells? Yes, broadly, two main types based on what they secrete. Serous cells produce watery secretions rich in non-glycosylated proteins like digestive enzymes.
Serous means protein-rich, watery.
Generally, yes. These cells typically have abundant rough ER for protein synthesis, a prominent Golgi for packaging, and distinct, often densely staining apical secretory granules. Think pancreas acinar cells or parotid salivary gland cells.
Okay. And the other type, mucus cells.
Mucus cells produce, well, mucus. They synthesize heavily glycosylated proteins called mucins. These mucins are stored in large, pale-staining apical granules called mucinogen granules.
Why pale staining?
Because mucins are very hydrophilic. During standard tissue preparation with aqueous solutions, they tend to swell up and wash out, leaving the apical cytoplasm looking empty or foamy. However, they stain intensely with PAS due to their high carbohydrate content. Goblet cells are the classic example, but you find mucus acini in salivary glands too.
Serous cells make proteins, mucus cells make mucins. Can glands have both?
Yes, some glands, like the submandibular salivary gland, are mixed seromucus, containing both types of cells. Sometimes you even see serous cells forming little caps, called serous demilines, around mucus acini.
Right. And one last cell type associated with glands, those contractile ones.
The myoepithelial cells, yes. These aren't secretory themselves, but they are essential for moving the secretions out.
Where are they found?
They sit between the secretory cells, serous or mucus, and the basement membrane in many exocrine glands, like sweat glands, mammary glands, salivary glands, lacrimal glands.
And they contract. How?
They look somewhat star-shaped or spindle-shaped and contain abundant actin filaments and myosin, the same proteins used in muscle contraction. They wrap their processes around the secretory units, acini or tubules.
Like squeezing a tube of toothpaste.
That's a great analogy. When the myoepithelial cells contract, they squeeze the secretory unit, increasing the pressure inside and helping to expel the accumulated secretions into the duct system quickly and efficiently.
Very clever. Okay, let's wrap up with some dynamic aspects. Transport across epithelia seems key.
Absolutely vital. Epithelia form barriers, but they also have to control what gets across. We talked about the transcellular pathway movement through the cell.
This often involves active transport.
Yes, especially for ions in water. Cells use specific pumps, like the Na plus K plus ATPase, usually located on the basolateral membrane, to move ions against their concentration gradients. Water often follows osmotically, sometimes facilitated by water channels called aquaporins.
And the tight junctions are crucial here.
They're essential to prevent the ions that were just pumped out from simply leaking back between the cells, paracellular backflow. They ensure the transport is directional and regulated.
You mentioned the kidney tubules earlier as an example of high energy demand for this.
Yes, the proximal tubule cells are masters of transcellular transport, reabsorbing salts, glucose, amino acids, and water. Their basal membranes are incredibly folded.
Increasing surface area.
Exactly. And packed right into those folds are huge numbers of mitochondria, churning out the ATP needed to power all the transport pumps. It's a beautiful correlation of structure and function reflecting the massive energy cost.
Okay, that's transcellular. What about transcytosis?
Transcytosis is different. It's about moving larger molecules or bulk fluid across the cell using vesicles.
How does that work?
Material is taken up on one side of the cell by endocytosis, packaged into vesicles, transported across the cytoplasm, and then released on the other side by exocytosis.
Like a ferry service across the cell.
Kind of, yeah. It's common in very thin cells, like the endothelium lining capillaries, allowing proteins and antibodies to move between blood and tissues, for example. It's less specific than channel or pump-mediated transport.
Got it. Now, epithelial cells don't last forever, do they? Renewal is important.
Extremely important. Most epithelia are constantly being worn away or damaged, so they need continuous renewal. We call them labile tissues.
How quickly are they replaced?
It varies enormously. The lining of your small intestine turns over completely in about 5 to 7 days. Skin takes longer, maybe a month. Cells in large glands like the liver or pancreas turn over very slowly, unless there's injury.
Where do the new cells come from? Stem cells.
Yes, adult stem cells are responsible for this renewal. Their location depends on the tissue type.
In stratified epithelia, like skin.
The stem cells and essentially all the mitotic activity are restricted to the basal layer, the layer sitting directly on the basement membrane. As cells divide, the daughter cells are pushed upwards and start differentiating.
What about in simpler tissues like the intestine?
There, the stem cells are usually found in protected niches. In the intestine, they're located down at the base of the crypts, the glands between the villi. In skin, stem cells reside in the basal layer and also in niches within hair follicles.
So specific locations are reservoirs for new cells, and this allows for repair.
Absolutely. Epithelia generally have a remarkable capacity for repair after injury. Even the liver, where mitosis is normally rare, can undergo massive regeneration if part of it is removed, driven by its epithelial-derived hepatocytes. The stem cells and the remaining differentiated cells can ramp up division.
Okay, finally, let's touch on pathology. What happens when epithelial growth goes wrong? Cancer.
Yes. Uncontrolled proliferation of epithelial cells is the basis of most common cancers. Before true cancer develops, we often see abnormal growth patterns called dysplasia, which might be precancerous and sometimes reversible.
But if it progresses?
If the abnormal growth becomes invasive and uncontrolled, it's neoplasia, or cancer. Malignant tumors arising from surface or lining epithelial tissue are broadly called carcinomas.
Carcinomas, like squamous cell carcinoma from stratified squamous epithelium.
Exactly. Or basal cell carcinoma, also from skin.
And if the cancer arises from glandular epithelium?
Then it's called an adenocarcinoma. Think colon cancer, breast cancer, prostate cancer, lung adenocarcinoma. These are all incredibly common, especially in adults over 45. They represent the vast majority of human cancers.
Derived from those secretory epithelial cells. And things like the loss of adherens we discussed are key steps in them becoming malignant.
Yes, disruption of cell adhesion, changes in polarity, alterations in basement membrane interactions. These are all critical events in the progression from normal epithelium to invasive carcinoma or adenocarcinoma.
Wow. We've covered a huge amount. From the molecules like laminin and claudin holding things together, to the energy demands of kidney transport, the different ways glands secrete, and how it all relates to health and disease. Epithelium is far more than just a simple covering.
It truly is. It's the dynamic, selective, regulatory interface between the body's internal environment and the outside world, or between different internal compartments. Structure and function are completely intertwined at every level.
Which brings us to a final thought. We've emphasized how utterly dependent these avascular epithelial cells are on diffusion from the connective tissue below across that basement membrane.
Absolutely critical lifeline.
So let's consider pathology impacting that lifeline. Imagine a chronic disease state, like long-term diabetes or hypertension, causes the basement membrane to gradually thicken and stiffen, making diffusion harder.
A known complication, yes. Reduced diffusion capacity.
Okay, thinking about all the high-energy epithelial processes, we discussed things like the massive active ion transporting kidney tubules, or the incredibly rapid cell renewal needed in the intestinal lining. Which of these high-demand functions do you think would be most vulnerable, most likely to fail first, if that basement membrane barrier becomes compromised and nutrient delivery slows down? And why? Something for you, the listener, to really think about.